Chapter 5 Molecular Basis of Inheritance. In the previous chapter, you have learned the inheritance pattern and the genetic basis of such patterns. At the time of Mendel, the nature of those factors regulating the pattern of inheritance was not clear. Over the next 100 years, the nature of the putative genetic material was investigated, culminating in the realization that DNA deoxyribonucleic acid is the genetic material, at least for the majority of organisms. In the class 11th, you have learnt that nucleic acids are polymers of nucleotide. Deoxyribonucleic acid DNA and the ribonucleic acid RNA are the two types of nucleic acids found in living system. The DNA acts as the genetic material in most of the organism. RNA through it also act as a genetic material in some viruses. Mostly functions as a messenger. RNA has additional roles as well. It functions as adapter structural and in some cases as a catalytic molecule. In class 11th you have already learned the structure of nucleotides and the way these monomer units are linked to form nucleic acid polymers. In this chapter, we are going to discuss the structure of DNA. In replication, the process of making RNA from DNA, transcription, the genetic code that determines the sequence of amino acids in proteins, the process of protein synthesis, translation, and elementary basis of their regulation, the determination of complete nucleotide sequence of human genome during last decade has set in a new era of genomics. In the last section, the essentials of human genome sequencing and its consequences will also be discussed. Let us begin our discussion by first understanding the structure of the most interesting molecule in the living system, that is the DNA. In subsequent sections, we will understand that why it is the most abundant genetic material and why its relationship is with RNA. 5.1 The DNA. DNA is a long polymer of deoxyribonucleotides. The length of DNA is usually defined as number of nucleotides or a pair of nucleotide referred to as base pairs present in it. This also is the characteristics of an organism. For example, a bacteriophage known as phi into 174 has 5386 nucleotides. Bacteriophage lambda has 48,502 base pairs. E. coli has 4.6 into 10's power 6 base pair, and haploid content of human DNA is 3.3 in 10's power 9 base pair. Let us discuss the structure of such a long polymer. 5.1.1 Structure of polynucleotide chain. Let us recapitulate the chemical structure of a polynucleotide chain, DNA or RNA. A nucleotide has three components: a nitrogenous base, a pentose sugar, ribose in case of RNA, and deoxyribose for DNA, and a phosphate group. There are two types of nitrogenous bases: purines, adenine, guanine, and pyrimidines, cytosine, uracil, and thymine. Cytosine is common for both DNA and RNA, and thymine is present in DNA. Uracil is present in RNA at the place of thymine. A nitrogenous base is linked to the OH of first carbon pentose sugar through a and glycosidic linkage to form a nucleoside, such as adenosine or deoxyadenosine, guanosine or deoxyguanosine, cytidine or deoxycytidine, and uridine or deoxythymidine. When a phosphate group is linked to OH of fifth carbon of a nucleoside through phosphodiester linkage or corresponding nucleotide or deoxynucleotide depending upon the type of sugar present is formed. Two nucleotides are linked through 3' to 5' phosphodiester linkage to form a dinucleotide. More nucleotides can be joined in such a manner to form a polynucleotide chain. A polymer thus formed has at one end a free phosphate moiety at 5' end of sugar, which is referred to as 5' end of polynucleotide chain. Similarity, at the other end of the polymer, the sugar has a free OH of 3' carbon group, which is referred to as 3' end of the polynucleotide chain. The backbone of a polynucleotide chain is formed due to sugar and phosphates. The nitrogenous base is linked to sugar moiety project from the backbone. Figure 5.1. In RNA, every nucleotide residue has an additional OH group present at second position in the ribose. Also in RNA, the uracil is found at the place of thymine, 5-methyl uracil, another chemical name for thymine. DNA as an acidic substance present in nucleus was first identified by Frederick Mischer in 1869. He named it as nuclein. However, due to technical limitation in isolating such a long polymer intact, the elucidation of structure of DNA remained elusive for a very long period of time. It was only in 1953 that James Watson and Francis Crick, based on the X-ray diffraction data produced by Maurice Wilkins and Rosalind Franklin, proposed a very simple but famous double helix model for the structure of DNA. One of the hallmarks of their propositions was base pairing between the two strands of polynucleotide chain. However, this proposition was also based on the observation of Irvin Chargaff's that for a double-stranded DNA, the ratio between adenine and thymine and guanine and cytosine was constant and equal 1. The base pairing confers a very unique property of the polynucleotide chains. They are said to be complementary to each other, and therefore, if the sequence of bases in one strand is known, then the sequence on other strand can be predicted. Also, if each strand from a DNA, let us call it as a parental DNA, act as a template for the synthesis of new strand, the two double stranded DNA, let us call them as daughter DNA, thus produced would be identical to the parental DNA molecule. Because of this, the genetic implications of the structure of DNA become very clear. The salient features of the double helix structure of DNA are as follows. First, it is made of two polynucleotide chain where the backbone is constituted by sugar phosphate and the bases projects inside. The two chains have anti-parallel polarity. It means if one chain has the polarity 5' to 3' and the other has 3' to 5'. 3. The bases in two strands are paired through hydrogen bond H bonds, forming base pairs. Adenine forms two hydrogen bonds with thymine form opposite strands and vice versa. Similarly, guanine is bonded with cytosine with three hydrogen bonds. As a result, always a purine comes opposite to a pyrimidine. This generates approximately uniform distance between the two strands of the helix. Figure 5.2. Fourth, the two chains are coiled in a right-handed fashion. The pitch of the helix is 3.4 nm. A nanometer is 1 billionth of a meter, that is 10's power 9 meter, and there are roughly 10 base pair in each turn. Consequently, the distance between a base pair in a helix is approximately 0.34 nm. The plane of one base pair stacks over the other in double helix. This, in addition to hydrogen bonds, confers the stability of helical structure. Figure 5.3. Compare the structure of purines and pyrimidines. Can you find out why the distance between two polynucleotide chains in DNA remains almost constant? The propositions of a double helix structure for DNA and its simplicity in explaining the genetic implications become revolutionary. Very soon, Francis Crick proposed the central dogma in molecular biology, which states that the genetic information flows from DNA to RNA and RNA to protein. Central dogma. In some viruses, the flow of information is in reverse direction, that is from RNA to DNA. Can you suggest a simple name to this process? 5.1.2 Packaging of DNA helix. Taken the distance between two consecutive base pairs as 0.34 nm, 0.34 multiply 10's power 9 meter. If the length of DNA double helix in a typical mammalian cell is calculated simply by multiplying the total number of base pair with distance between two consecutive base pair, that is 6.6 multiply 10's power 9 base pair multiply 0.34 multiply 10's power minus 9 meter per base pair, it comes out to be approximately 2.2 meters, a length that is far greater than the dimensions of a typical nucleus, approximately 10's power 6 meter. How is such a long polymer packed in a cell? If the length of E. coli DNA is 1.36 millimeter, can you calculate the number of base pairs in E. coli? In prokaryotes such as E. coli, though they do not have a defined nucleus, the DNA is not scattered throughout the cell. DNA being negatively charged is held with some proteins that have positive charges in the region termed as nucleoid. The DNA in nucleoid is organized in large group held by proteins. In eukaryotes, this organization is much more complex. There is a set of positively charged basic proteins called histones. A protein acquires charge depending upon the abundance of amino acid residue with charged side chains. Histones are rich in basic amino acid residues lysine and arginine. Both the amino acid residues carry positive charges in their side chain. Histones are organized to form a unit of 8 molecules called histone octamer. The negatively charged DNA is wrapped around the positively charged histone octamer to form a structure called nucleosome, figure 5.4a. A typical nucleosome contains 200 base pairs of DNA. Nucleosome constitutes the repeating unit of a structure in nucleus called chromatin, thread-like stained colored body seen in nucleus. The nucleosomes in chromatin are seen as beads on string structure when viewed under electron microscope EM, figure 5.4b. Theoretically, how many such beads nucleosomes do you imagine are present in mammalian cell? Question mark. The beads on string structure in chromatin is packed to form chromatin fiber that are further coiled and condensed at metaphasic state of cell division to form chromosome. The packaging of chromatin in higher level requires additional set of proteins that are collectively referred as non-histone chromosomal NHC proteins. In a typical nucleus, some regions of chromatin are loosely packed and stains light and are referred to as euchromatin. The chromatin that is more densely packed and stains dark are called heterochromatin. Euchromatin is said to be transcriptionally active chromatin, whereas heterochromatin is inactive. 5.2 Search for genetic material. Even though the discovery of nuclein by Miescher and the propositions for principles of inheritance by Mendel was almost at the same time, but that the DNA acts as a genetic material took long to be discovered and proven. By 1926, the quest to determine the mechanism for genetic inheritance had reached the molecular level. The previous discoveries by Gregor Mendel, Walter Sutton, Thomas Hunt Morgan, and numerous other scientists had narrowed the search to the chromosomes located in the nucleus of most cells. But the question of what molecules was actually is genetic material had not been answered. Transforming principle. In 1928, Frederick Griffith, in a series of experiments with Streptococcus pneumoniae, a bacterium responsible for pneumonia, witnessed a miraculous transformation in the bacteria. During the course of his experiment, a living organism, bacteria, had changed in physical form. When Streptococcus pneumoniae, pneumococcus bacteria are grown on a culture plate, some produce smooth, shiny colonies S, while other produce rough colonies R. This is because the S strain bacteria have a mucous polysaccharide coat, while R strain does not. Mice infected with the S strain virulent die with pneumonia infection, but mice infected with the R strain do not develop pneumonia. S strain inject into mice, mice die. R strain inject into mice, mice live. Griffith also able to kill bacteria by heating them. He observed that heat killed S strain bacteria injected into mice did not kill them. When he S strain heat killed inject into mice live. S killed heat strain plus R strain live inject into mice die. Injected a mixture of heat killed S and live R bacteria, the mice died. Moreover, he recovered living S bacteria from the dead mice. He concluded that the R strain bacteria had somehow been transformed by the heat killed S strain bacteria. Some transforming principle transformed from the heat killed S strain had enabled the R strain to synthesize a smooth polysaccharide coat and become virulent. This must be due to the transfer of the genetic material. However, the biochemical nature of genetic material was not defined from his experiments. Biochemical characterization of transforming principle. Prior to the work of Oswald Overy, Colin MacLeod, and Macklin McCarthy, 1933 44, the genetic material was thought to be a protein. They worked to determine the biochemical nature of transforming principles in Griffith experiment. They purified biochemicals, proteins, DNA, RNA, etc. from the heat-killed S cell to see which one could transform live R cell into S cell. They discovered that DNA alone from S bacteria caused R bacteria to become transformed. They also discovered that protein digesting enzyme proteases and RNA digesting enzyme RNAs did not affect transformation, so the transforming substance was not a protein or RNA. Digestion with DNAs did inhibit transformation, suggesting that the DNA caused the transformation. They concluded that DNA is the hereditary material, but not all biologists were convinced. Can you think if any difference between DNA and DNAs? 5.2.1, the genetic material is DNA. The unequivocal proof that DNA is the genetic material came from the experiment of Alfred Hershey and Martha Chase in 1952. They worked with viruses that infect bacteria called bacteriophages. The bacteriophage attack to the bacteria and its genetic material then enters the bacterial cell. The bacterial cell treats the viral genetic material as if it was its own and subsequently manufactures more virus particles. Hershey and Chase worked to discover whether it was protein or DNA from the virus that entered the bacteria. They grew some virus in a medium that contained radioactive phosphorus and some other on medium that contained radioactive sulfur. Viruses grown in the presence of radioactive phosphorus contained radioactive DNA but not radioactive protein because DNA contains phosphorus but protein does not. Similarly, viruses grown on radioactive sulfur contain radioactive protein but not radioactive DNA because DNA does not contain sulfur. Radioactive phases were allowed to attach to E. coli bacteria. Then, as the infection proceeded, the viral coats were removed from the bacteria by agitating them in a blender. The virus particles were separated from the bacteria by spinning them in a centrifuge. Bacteria which was infected with viruses that had radioactive DNA were radioactive, indicating that DNA was the material that passed from the virus to the bacteria. Bacteria that were infected with virus that had radioactive proteins were not radioactive. This indicates that proteins did not enter the bacteria from the viruses. DNA is therefore the genetic material that is passed from virus to bacteria, figure 5.5. 5.2.2, Properties of genetic material, DNA versus RNA. From the foregoing discussion, it is clear that the debate between proteins versus viruses as the genetic material was unequivocally resolved from Hershey-Chase experiment. It became an established fact that it is DNA that act as genetic material. However, it subsequently became clear that in some viruses, RNA is the genetic material. For example, tobacco mosaic virus, QB bacteriophage, etc. Answer to some of the questions such as why DNA is the predominant genetic material, whereas RNA performs dynamic function of messenger and adapter, has to be found from the differences between chemical structures of the two nucleic acid molecules. Can you recall the two chemical differences between DNA and RNA? A molecule then can act as a genetic material must fulfill the following criteria. First, it should be able to generate its replica replication. 2, it should be stable chemically and structurally. 3, it should provide the scope for new changes mutation that are required for evolution. Fourth, it should be able to express itself in the form of Mendelian characteristics. If you examine such if you examine each requirement one by one because of role of base pairing and complementarity, both the nucleic acid DNA and RNA have the ability to direct their duplications. The other molecules in the living system, such as proteins, fulfill first criteria itself. The genetic material should be stable enough not to change with different stages of life cycle, age, or with change in philosophy of the organism. Stability as one of the properties of genetic material was very evident in Griffith's transforming principle itself, that heat which killed the bacteria at least did not destroy some of the properties of genetic material. This now can easily be explained in light of the DNA, that the two strands being complementary separated by heating come together when appropriate conditions were provided. Further, 2' OH group present at every nucleotide in RNA is a reactive group and make RNA labile and easily degradable. RNA is also now known to be catalytic, hence reactive. Therefore, DNA chemically is less reactive and structurally more stable when compared to RNA. Therefore, among the two nucleic acid, the DNA is a better genetic material. In fact, the presence of thymine at the place of uracil also confers additional stability of DNA. Details discussion about the requires understanding of the process of repair in DNA, and you will study these processes in higher classes. Both DNA and RNA are able to mutate. In fact, RNA being unstable mutate at a faster rate. Consequently, viruses having RNA genome and having shorter lifespan mutate and evolve faster. RNA can directly code for the synthesis of proteins, hence can easily express the characters. DNA, however, is dependent on RNA for synthesis of proteins. The protein synthesizing machinery has evolved around RNA. The above discussion indicate that both RNA and DNA can function as genetic material, but DNA being more stable is preferred for storage of genetic information. For the transmission of genetic information, RNA is better. 5.3 RNA word. From foregoing discussion, an immediate question becomes evident: which is the first genetic material? It shall be discussed in detail in the chapter on chemical evolution, but briefly we shall highlight some of the facts and points. RNA was the first genetic material. There is now enough evidence to suggest that essential for life processes such as metabolism, translation, splicing, etc., evolved around RNA. RNA used to act as a genetic material as well as a catalyst. There are some important biochemical reactions in living system that are catalyzed by RNA catalyst and not by protein enzymes. But RNA being a catalyst was reactive and hence unstable. Therefore, DNA has evolved from RNA with chemical modifications that make it more stable. DNA being double stranded and having complementary strand further resists changes by evolving a process of repair. 5.4 Replication. While proposing the double helical structure for DNA, Watson and Crick had immediately proposed a scheme for replication of DNA to create their original statement that is as follows. It It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible coping mechanism for the genetic material. Watson and Crick, 1953. The scheme suggested that the two strands would separate and act as a template for the synthesis of new complementary strands. After the completion of replication, each DNA molecule would have one parental and one newly synthesized strand. This scheme was termed as semi-conservative DNA replication, figure 5.6. 5.4.1, the experimental proof. It is now patterned that DNA replicates semi-conservatively. It was shown first in Escherichia coli and subsequently in higher organisms such as plants. The human cells. Matthew Messelson and Franklin Stahl performed the following experiment in 1958. First, they grow E. coli in a medium containing 15 ammonium chloride. 15 nitrogen is a heavy isotope of nitrogen as the only nitrogen source for many generations. The result was that N15 was incorporated into newly synthesized DNA as well as other nitrogen containing compounds. This heavy DNA molecule could be distinguished from the normal DNA by centrifugation in a cesium chloride density gradient. Please note that N15 is not a radioactive isotope and it can be separated from N14 only by densities. Number two, then they transformed the cells into a medium with normal ammonium chloride and took samples at various definite time intervals as the cell multiplied and extracted the DNA that remained as double stranded helices. The various examples were separated independently on cesium chloride gradients to measure the densities of DNA, figure 5.7. Can you recall what centrifugal force is and think why a molecule with higher mass density would sediment faster? The results are shown in figure 5.7. Third, thus the DNA that was extracted from the culture one generation after the transfer from N15 to N14 medium, that is after 20 minutes, E. coli divides in 20 minutes, had a hybrid or immediately densities. DNA extracted from the culture after another generation, that is after 40 minutes, second generation, was composed of two equal amounts of the hybrid DNA and of light DNA. If E. coli was allowed to grow for 80 minutes, then what would be the proportions of light and hybrid density DNA molecules? With similar experiments involving use of radioactive thymidine to detect distribution of newly synthesized DNA in the chromosome was performed on Vicia fava beans by Taylor and the colleagues in 1958. The experiment proved that the DNA in chromosome also replicates semi-conservatively. 5.4.2, the machinery and the enzymes. In living cells such as E. coli, the process of replication requires a set of catalysts enzymes. The main enzyme is referred to as the DNA dependent DNA polymerase since it uses a DNA template to catalyze the polymerization of deoxynucleotides. These enzymes are highly effective enzymes as they have to catalyze polymerization of a large number of nucleotides in a very short time. E. coli that has only 4.6 into 10's power 6 base pair compared it with human those deployed contain is 6.6 multiply 10's power 9 base pair completes the process of replication within 18 minutes. That means the average rate of polymerization has to be approximately 2000 base pair per second. Not only do these polymerases have to be fast, but they also have to catalyze the reaction with high degree of accuracy. Any mistake during replication would result into mutation. Furthermore, energetically replication is a very expensive process. Deoxyribonucleoside triphosphates serves dual purposes. In addition to acting as substrates, they provide energy for polymerization reaction. The two terminal phosphates in the deoxyribonucleoside triphosphate and high energy phosphate, same as in case of ATP. In addition to DNA dependent DNA polymerases, many additional enzymes are required to complete the process of replication with high degree of accuracy. For long DNA molecules, since the two strands of DNA cannot be separated in its entire length due to very high energy requirement. The replication occurs within a small opening of the DNA helix referred to as replication fork. The DNA dependent DNA polymerases catalyzes polymerization only in one direction, that is 5 prime to 3 prime. This creates some additional complications in the replicating fork. Consequently, on one strand, the template with polarity 3 prime to 5 prime, the replication is continuous, while on the other, the template with polarity 5.23 prime, it is discontinuous. The discontinuously synthesized fragments are later joined by enzyme DNA ligase, figure 5.8. The DNA polymerases on their own cannot initiate the process of replication. Also, the replication does not initiate randomly at any place in DNA. There is a definite reason in E. coli DNA where the replication originates. Such origins is termed as origin of replication. It is because of the requirement of the origin of replication that a piece of DNA, if needed to be propagated during recombinant DNA procedures, requires a vector. The vectors provide the origin of replication. Further, not every detail of replication is understood well. In eukaryotes, the replication of DNA takes place in S phase of the cell cycle. The replication of DNA and cell division cycles should be highly coordinated. If failure in cell division after DNA replication results into polyploidy, a chromosomal abnormality, you will learn the detailed nature of origin and the processes occurring at this site in higher classes. 5.5 Transcription. The process of copying genetic information from one strands of the DNA into RNA is termed as transcription. Here also the principle of complementarity governs the process of transcription, except the adenosine complements now forms base pair with uracil instead of thymine. However, unlike in the process of replication, which once set in the total DNA of an organism get duplicated, in transcription, only a segment of DNA and only one of the strand is copied into RNA. This necessitates defining the boundaries that would demarcate the region and the strand of DNA that would be transcribed. Why both these strands are not copied during transcription has the simple answer. First, if both strands act as template, they would code for RNA molecule with different sequences. Remember, complementarity does not remain identical, and in turn, if they code for proteins, the sequence of amino acid in the protein would be different. Hence, one segment of the DNA would be coding for two different proteins, and this would complete the genetic information transfer machinery. Second, the two RNA molecules, if produced simultaneously, would be complementary to each other, hence would form a double-stranded RNA. This would prevent RNA from being translated into protein, and the exercise of transcription would become a futile one. 5.51 Transcription unit. A transcription unit in DNA is defined primarily by the three regions in the DNA: a promoter, 2 the structural gene, and 3 the terminator. There is a convention in defining the two strands of the DNA in the structural gene of a transcription unit. Since the two strands have opposite polarity and the DNA dependent RNA polymerase also catalyze the polymerization in only one direction, that is 5 prime to 3 prime. The strand that has the polarity 3 prime to 5 prime act as template and is also referred to as template strand. The other strand which has the polarity 5 prime to 3 prime and the sequence same as RNA except thymine in place of uracil is displaced during transcription. Strangely, this strand which does not code for anything referred to as coding strand. All the reference point while defining a transcription unit is made with coding strand. To explain the point, a hypothetical sequence from a transcriptional unit is represented below: 3 prime ATGC 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 5 prime template strand, 5 prime TACG 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 3 prime coding strand. Can you Now write the sequence of RNA transcribed from the above DNA. The promoter and terminator flank the structural gene in a transcription unit. The promoter is said to be located towards 5' and upstream of the structural gene. The reference is made with respect to the polarity of coding strand. It is a DNA sequence that provides binding site for RNA polymerase, and it is the presence of a promoter in a transcription unit that also defines the template and coding strands. By switching its position with terminator, the definition of coding and template strands would be reversed. The terminator is located towards 3' and downstream of the coding strand and is usually defined the end of the process of transcription of figure 5.9. There are additional regulatory sequences that may be present further upstream or downstream to the promoter. Some of the properties of these sequences shall be discussed while dealing with regulation of gene expression. 5.5.2 Transcription unit and the gene. A gene is defined as the functional unit of inheritance, though there is no ambiguity that the genes are located on the DNA. It is difficult to literally define a gene in terms of DNA sequence. The DNA sequence coding for transfer RNA or RNA molecule also define a gene. However, by defining a cistron as a segment of DNA coding for a polypeptide, the structural gene in a transcription unit could be said as monocistronic, mostly in eukaryotes, or polycistronic, mostly in bacteria or prokaryotes. In eukaryotes, the monocistronic structural genes have interrupted coding sequences. The genes in eukaryotes are split. The coding sequences are or express the sequences are defined as exons. Exons are said to be those sequences that appear in mature or processed RNA. The exons are interrupted by introns. Introns are intervening sequences do not appear in mature or processed RNA. The split gene arrangement further complicates the definition of a gene in terms of a DNA segment. Inheritance of a character is also affected by promoter and regulatory sequences of a structural gene. Hence, sometimes the regulatory sequences are loosely defined as regulatory genes, even though these sequences do not code for any RNA or protein. 5.5.3 Types of RNA and the process of transcription. In bacteria, there are three major types of RNAs: mRNA, messenger RNA, tRNA, transfer RNA, rRNA, ribosomal RNA. All these RNA are needed to synthesize a protein in a cell. mRNA provides the template, tRNA begins amino acids and read the genetic code, and rRNA plays structural and catalytic role during translation. There is single DNA dependent RNA polymerase that catalyzes transcription in all type of RNA in bacteria. RNA polymerase bind to promoter and initiate transcription initiation. It uses nucleoside triphosphate as substrate and polymerizes in a template dependent fashion following a rule of complementarity. If somehow also facilitates opening of a helix and continue elongation. Only a short stretch of RNA remains bound to the enzyme. Once the polymerase reaches the terminator region, the nascent free RNA falls off, so also the RNA polymerase. The result is termination of transcription. An intriguing question that is, how is the RNA polymerase is able to catalyze all the three steps, which are initiation, elongation, and termination? The RNA polymerase is only capable of catalyzing the process of elongation. It associate transiently with initiation factor sigma and terminator factor rho to initiate and terminate the transcription respectively. Association with these factors alter the specificity of the RNA polymerase to either initiate or terminate, figure 5.10. In bacteria, since the mRNA does not require any processing to become active, and also since transcription and translation takes place in the same compartment, there is no separation of cytosol and nucleus in bacteria. Many times the translation can begin much before the mRNA is fully transcribed. Consequently, the transcription and translation can be copied in bacteria. In eukaryote, there are two additional complexities. First, there are at least three RNA polymerases in the nucleus. In addition to the RNA polymerase found in the organelle, there is a clear-cut division of labor. The RNA polymerase first transcribes rRNA 20S, 18S, and 5.8S, whereas the RNA polymerase third is responsible for transcription of tRNA, 5S RNA, and snRNA, small nuclear RNAs, and RNA polymerase second transcribes precursor of mRNA, the heterogeneous nucleus RNA, hnRNA. Second, the second complexity is that the primary transcripts contains both the exons and the introns and are non-functional. Hence, it is subjected to the process called splicing, where the introns are removed and exons are joined to a defined order. hnRNA undergoes additional processing called as capping and tailing. In capping, an unusual nucleotide methylguanosine triphosphate is added to the 5' end of hnRNA. In tailing, adenylate residues 200 to 300 are added at a 3' end of the template independent manner. It is fully processed hnRNA, now called mRNA, that is transcribed out of the nucleus for translation, figure 5.11. The significance of such complexity is now begun to be understood. The split gene arrangement represent probably an ancient features of the genome. The presence of introns is reminiscent of antiquity, and the process of splicing represent the dominance of RNA world. In recent times, the understanding of RNA and RNA dependent processes in the living system have used more importance. 5.6 genetic codes during replication, transcription, and nucleic acid was copied to form another nucleic acid. Hence, these processes are easy to conceptualize on the basis of complementarity. The process of translation requires transfer of genetic information from a polymer of nucleotides to synthesize a polymer of amino acids. Neither does any complementarity exist between nucleotides and amino acids, nor could any be drawn theoretically. There existed ample evidences through to support the notion that changes in nucleic acid genetic material were responsible for change in amino acids in proteins. This led to the proposition of the genetic code that could direct the sequence of amino acid during synthesis of proteins. If determining the biochemical nature of genetic material and the structure of DNA was very exciting, the proposition and dissipating of genetic code over most challenging. It in a very true sense, it required involvement of scientists from several disciplines, psychists, organic chemist, biochemist, and geneticists. It was George Gamow, a psychist, who argued that since there are only four bases and if they have to code for 20 amino acids, the code should constitute a combination of bases. He suggested that in order to code for all the 20 amino acids, the code should be made up of three nucleotides. This would this was a very bold proposition because a permutation combination of force power 3 4 multiplied by 4 multiplied by 4 would generate 64 codons, generating many more codons than required. Providing proof that the codon was a triplet was a more denuding task. The chemical method developed by Hargobind Khurana was instrumental in synthesizing RNA molecule with defined combination of bases, homopolymers and copolymers. Marshall Nierenberg's cell-free system for protein synthesis finally helped the code to be dissipated. Severo-OCHA enzyme polynucleotide phosphorylase was also helpful in polymerizing RNA with defined sequences in a template-independent manner. Enzyme synthesis of RNA, finally, a cracker board for genetic code was predicted, which is given in table 5.1. The salient feature of genetic codes are as follow. First, the codon is triplet. 61 codons code for amino acid and 3 codons do not code for any amino acid. Hence, they function as stop codons. Number 2, some amino acids are coded by more than one codon. Hence, the code is degenerate. Number 3, the codon is read in mRNA is a contiguous fashion. There are no punctuations. Number 4, the code is nearly universal. For example, from bacteria to human, UUU would code for phenylalanine, PHE. Some exceptions to this rule would have been found in mitochondrial codons and in some protozoans. Fifth, AUG has dual functions as code for methionine MET, and it also acts as initiator codon. Sixth, UAA, UAG and UGA are stop terminator codon. If following is a sequence of nucleotides in mRNA, predict the sequence of amino acid coded by it, take help of the checkerboard. Now try the possible. Following is the sequence of amino acid coded by an mRNA. Predict the nucleotide sequence in the mRNA. Do you face any difficulty in predicting the opposite? Can you now correlate which two properties of genetic code you have learnt? 5.6.1 Mutations and genetic code. The relationship between genes and DNA are best understood by mutation studies. Have you studied about mutation and its effect in chapter 4? Effects on large deletions and rearrangement in a segment of DNA are easy to comprehend. It may result in loss or gain of a gene and as a function. The effect of point mutations will be explained here. A classical example of point mutation is a change of single base pair in a gene for beta globin chain that results in the change of amino acid residue glutamate to valine. It results into the diseased condition called a sickle cell anemia. Effect of point mutation that inserts or deletes a base in structural gene can be better understood by following simple example. Consider a statement that is made up of the following words such as having three letters like genetic code. Ram has red cap. If we insert a letter B in between has and red and rearrange the statement, it would read as follows: Ram has BREDCAP. Similarly, if we now insert two letters at the same place, say BI, now it would read Ram has BIREDCAP. Now we insert three letters together, say big, the statement would be Ram has big red cap. The sum exercise can be repeated by deleting the letters RED one by one and rearranging these statements to make a triplet word. Ram has EDCAP, Ram has DCAP, Ram has CAP. The conclusion from the our exercise is very obvious. Insertion or deletion of two or more base changes the reading frame from the point of insertion or deletion. However, such mutation are referred to as frame shift insertion or deletion mutation. Insertion or deletion of three or its multiple bases insert or delete in one or multiple codon, hence one or multiple amino acid, and reading frame remains unaltered from that point onward. 5.6.2 tRNA at the adapter molecule. From the very beginning of the propositions of code, it was clear to Francis Crick that there had to be a mechanism to read the code and also to link it to the amino acid, because amino acids have no structural specialties to read the code uniquely. He postulated the presence of an adapter molecule that would on one hand read the code and on other hand would bind to specific amino acids. The tRNA, then called sRNA, soluble RNA, was known before the genetic code was postulated. However, its role as an adapter molecule was assigned much later. tRNA has an anticodon loop that has bases complementary to the code, and it's had an amino acid acceptor and to which it binds to amino acid. tRNA are specific for each amino acid, figure 5.12. For initiation, there is another specific tRNA that is referred to as initiator tRNA. There are no tRNA for stop codons. In figure 5.12, the secondary structure of tRNA has been depicted that looks like a clover leaf. The actual structure, the tRNA, is a compact molecule which look like inverted L, full stop. 5.7 Translation. Translation refers to the process of polymerization of amino acids to form a polypeptide, figure 5.13. The order and sequence of amino acids are defined by the sequence of bases in the mRNA. The amino acids are joined by a bond which is known as the peptide bond. Formation of a peptide bond requires energy. Therefore, in the first phase, at least itself amino acids are activated in the presence of ATP and linked to their cognate tRNA, a process commonly called as charging of tRNA or amino acylation of tRNA to be more specific. If two such charged tRNA are brought close enough, the formation of peptide bond between them would be favored energetically. The presence of a catalyst would enhance the rate of the peptide bond formation. The cellular factory responsible for synthesizing protein is the ribosome. The ribosome consists of structural RNA and about 80 different proteins. In its inactive state, it exists as two subunits, a large subunit and a small subunit. When the small subunit encounters an mRNA, the process of translation of the mRNA to protein begins. There are two sites in the large subunit and subsequent amino acid to bind and thus to close enough to each other for the formation of a peptide bond. The ribosome also acts as a catalyst 23 as RNA in bacteria as the enzyme ribosome for the formation of peptide bond. The translation unit in mRNA is the sequence of hRNA that is flanked by the start codon AUG and the stop codon and codes for a polypeptide. And mRNA also has some additional sequences that are not translated and referred to as untranslated region UTR. The UTR are present at both 5' ends before start codon and the 3' end after stop codon. They are required for efficient translation process. For initiation, the ribosomes bind to the mRNA to the start codon AUG that is recognized only by the initiator tRNA. The ribosome proceeds to the elongation phase to protein synthesis. During this stage, complexes composed of an amino acid linked to tRNA subsequently bind to the appropriate codon in mRNA by forming complementary base pair with the tRNA anticodon. The ribosomes move from codon to codon along the mRNA. Amino acids are added one by one, translated into polypeptide sequence detached by DNA and represented by mRNA. At the end, a release factor binds to the stop codon, terminating translation and releasing the complete polypeptide from the ribosome. 5.8 Regulation of gene expression. Regulation of gene expression refers to a very broad term that may occur at various levels. Considering that gene expression results in the formation of a polypeptide, it can be regulated at several levels. In eukaryotes, the regulation could be extracted at first transcriptional level, formation of primary transcript; second, processing level, regulating or splicing; third, transcription of mRNA from nucleus to the cytoplasm; from translation level. The genes in a cell are expressed to perform particular function or a set of functions. For example, if an enzyme called beta-galactosidase is synthesized by E. coli, it is used to catalyze the hydrolysis of a disaccharide lactose into galactose and glucose. The bacteria use them as a source of energy. Hence, if the bacteria do not have lactose around them to be utilized for energy source, they would no longer require the synthesis of the enzyme beta-galactosidase. Therefore, in simple terms, it is a metabolic, physiological, environmental conditions that regulate the expression of genes. The development and differentiation of embryo into adult organisms are also a result of the coordinated regulation of expression of several set of genes. In prokaryotes, control of the rate of transcriptional initiation is the predominant site for control of gene expression. In the transcription unit, the activity of RNA polymerase as a given promoter is in turn regulated by interaction with accessory protein which affects its ability to recognize start site. These regulatory proteins can act both positively activators and negatively repressor. The accessibility of promoter regions of prokaryotic DNA is in many cases regulated by the interaction of proteins with sequences termed operators. The operator regions in adjacent to the promoter elements in some operands, and in most cases the sequence of the operator bind a repressor protein. Each operon has its specific operator and specific repressor. For example, lac operator is present only in lac operon and it interacts specifically with lac repressor only. 5.81 Lac operon. The elucidation of the lac operon was also a result of a close association between the genetist Francis Jacob and a biochemist Jacob Monod. They were the first to elucidate the transcriptionally regulated system in lac operon. Here, lac refers to lactose, a polycistronic structural gene is regulated by a common promoter and regulatory genes. Such arrangement is very common in bacteria and is referred to as operon. To name such Example, lac operon, trap operon, ora operon, his operon, val operon, etc. The lac operon consists of one regulatory gene, that is the I gene. Here, the term I does not refer to inducer. Rather, it is derived from the word inhibitor. The three structural genes Z, Y, and A. The I gene codes for the repressor of the lacoperon. Z gene codes for the beta galactosidase B-gal, which is primarily responsible for the hydrolysis of the disaccharide lactose into its monomeric units galactose and glucose. The Y gene codes for the permease, which increases permeability of the cell to beta galactosidase. A gene A codes transacetylase. Hence, all the three gene products in lac-operon are required for metabolism of lactose. In most other operons as well, the gene present in an operon are needed altogether to function in the same or related metabolic pathway. Figure 5.14. Lactose is a substrate for the enzyme beta galactosidase and is regulates switching on and off of the lagon operon. Hence, it is named as inducer. In the absence of a preferred carbon source such as glucose, if lactose is provided in the growth medium of the bacteria, lactose is transported into the cell through the action of permease. Remember, a very low level of expression of lacoperon has to be present in cell all time. Otherwise, lactose cannot enter the cells. The lactose then induces the operon in a following manner. The repressor of the operon is synthesized all the time consecutively from the I gene. The repressor protein binds to the operator region of the operon and prevents RNA polymerase from transcribing the operon. In the presence of an inducer such as lactose or allolactose, the repressor is inactivated by interaction with the inducer. This allows RNA polymerase access to the promoter and transcription proceeds. Figure 5.14. Essentially, regulations of lamp cropron can also be visualized as regulation of enzyme synthesis by its substrate. Remember, glucose or galactose cannot access inducer for lac operon. Can you think for how long the lac operon would be expressed in the presence of lactose? Regulation of lac operon by repressor is referred to as negative operon. Lac operon is under the control of positive regulation as well, but it is beyond the scope of discussion at this level. 5.9 Human Genome Project. In the preceding sections, you have learned that it is the sequence of bases in the DNA that determines the genetic information of a given organism. In other words, genetic makeup of an organism or an individual lies in their DNA sequences. If two individuals differ, then their DNA sequences should also be different, at least at some places. These assumptions led to the quest of finding out the complete DNA sequence of human genome with the establishment of genetic engineering techniques where it was possible to isolate and clone any piece of DNA, and availability of simple and fast technique for determining DNA sequences. A very ambitious project of sequencing human genome was launched in the year 1990. Human Genome Project was called a mega project. You can imagine that magnitude and the requirements for the project if was we simply define the aims of the project as follow. Human genome is said to have approximately 3 multiplied 10 power 9 base pair, and if the cost of sequencing required in US dollar 3 per base pair, the estimated cost in the beginning, the total estimated cost of the project will be approximately 9 billion US dollar. Further, if the obtained sequences were to be stored in typed from in books, and if each page of a book contained 1000 letter and each book contained 1000 pages, then 3300 such books would be required to store the information of DNA sequence from a single human cell. The enormous amount of data expected to be generated also necessitate the use of high speed computational devices, the data source, the retrieval, and synthesis. GAT Human Genome Project was closely associated with the rapid development of a new era in biology called bioinformatics. Goals of Human Genome Project. Some of the important goals of Human Genome Project were as follows. First, identify all the approximately 20,000 to 25,000 genes in human DNA. Second, determine the sequence of the 3 billion chemical base pairs that make up human DNA. Third, store this information in database. Fourth, improve tools for data analysis. Fifth, transfer related technology to other sectors such as industries. Sixth, address the ethical, legal, and social issues (ELSI) that may arise from the project. The Human Genome Project was a 13 years project coordinated by the US Department of Energy and the National Institutes of Health. During the early years of the Human Genome Project, the Wellcome Trust UK became a major partner. Additional contributions came from Japan, France, Germany, China, and others. The project was completed in 2003. Knowledge about the facts of DNA variations among individuals can lead to revolutionary new ways to diagnose, treat, and someday prevent the thousands of disorders that affect human beings. Besides providing clues to understanding human biology, learning about non human organisms' DNA sequence can lead to an understanding of their natural capabilities that can be applied towards solving challenges in healthcare, agriculture, energy production, environmental remediation. Many non human model organisms, such as bacteria, yeast, C. elegans, a cell free living non pathogenic nematode, Drosophila, the fruit fly plants, rice, and Arabidopsis, etc., have also been sequenced. Methodologies. The method involved two major approaches. One approach focused on identifying all the genes that are expressed as RNA, referred to as expressed sequence text EST. The other took the blind approach to simply sequencing the whole set of genome that contained all the coding and non-coding sequence and later assigning different regions in the sequence with functions, a term referred to as sequence annotation. For sequencing, the total DNA from a cell is isolated and converted into random fragments to relatively smaller sizes. Recall DNA is a very long polymer and there are technical limitations in sequencing very long pieces of DNA and cloned in suitable host using specialized vector. The cloning resulted into amplification of each piece of DNA fragment so that it subsequently could be sequenced with ease. The commonly used hosts were bacteria and yeast, and the vectors were called as BAC, bacterial artificial chromosome, and YAC, yeast artificial chromosome. The fragments were sequenced using automated DNA sequencers that worked on the principle of the method developed by Frederick Singer. Remember, Singer is also credited for developing method for determination of amino acid sequences in proteins. These sequences were then arranged based on upon overlapping regions present on them. These required generations of overlapping fragments for sequencing. Alignment of those sequences was humanity not possible. Therefore, specialized computer-based programs were developed. Figure 5.15. These sequences were subsequently annotated and were assigned to each chromosome. The sequence of chromosome 1 was completed only in May 2006. This was the last of the 24 human chromosome, 22 autosome, and XY to be sequenced. Another challenging task was assigning the genetic and physical map on the genome. This was generated using information on polymorphism of restriction endonuclease recognition sites and some repetitive DNA sequences known as microsatellized. One of the applicants of polymerization is repetitive DNA sequence shall be explained in next section of DNA fingerprinting. 5.9.1 Salient features of human genome. Some of the salient observations drawn from human genome project are as follow. Number 1, human genome contains 3164.7 million base pairs. Number 2, the average gene consists of 3000 bases but size vary greatly, with the largest known human gene being dystrophin at 2.4 million bases. Third, the total number of genes is estimated at 30,000, much lower than previous estimates of 80,000 to 1,40,000 genes. Almost all 99.9% nucleotide bases are exactly the same in all humans. Number 4, the functions are unknown for over 50% of the discovered gene. Number 5, less than 2% of the genome codes for protein. Number 6, repeated sequence make up very large portion of the human genome. Number 6, Repetitive sequence are stretches of DNA sequences that are repeated many times, sometimes hundreds to thousand times. They are thought to have no direct coding functions, but they shed light on chromosome structure, dynamics, and evolution. Number 8, chromosome 1 has most gene 2986 and Y has the fewest 231. Nine scientists have identified about 1.4 million locations where single base DNA differences SNP single nucleotide polymorphism pronounced as SNPs occurs in human. This information promises the revolutionize the process of finding chromosomal locations for disease associated sequences and tracing human history. 5.9.2 applications and future challenges. Deriving meaningful knowledge from the DNA sequences will define research through the coming decades, leading to our understanding of biological system. The enormous task will require the expertise and creativity 2000 of scientists from varied disciplines in both the public and private sector worldwide. One of the greatest impacts of having the HG sequence and may well be enabling a radically new approach to biological research. In the past, research studied one or a few genes at a time. With whole genome sequences, the new high throughput technology, we can approach questions systematically and on a much broader scale. They can study all the genes in a genome, for example, all the transcript in a particular issue or organ or tumor, or how tens of thousands of genes and proteins work together in interconnected network to orchestrate the chemistry of life. 5.10 DNA fingerprinting. As stated in the preceding sector, 9.9% of base sequence among humans is the same, assuming human genome as 3.10 power 9 base pair is how many base sequences would there be differences. It is these differences in a sequence of DNA which make every individual unique in their phenotypic appearance. If one aims to find out genetic differences between two individual or among individual of a population, sequencing the DNA every time would be a daunting and expensive task. Imagine trying to compare two sets of 3 multiply 10 base power 10 6 base pairs. DNA fingerprinting is a very quick way to compare the DNA sequences of two individual. DNA fingerprinting involve identifying differences in some specific regions in DNA sequence called a repetitive DNA, because in these sequences a small stretch of DNA is repeated many times. These repetitive DNA are separated from bulk genomic DNA as different peaks during density gradient centrifugation. The bulk DNA forms a major peak and the other small peaks are referred to as a satellite DNA. Depending on base composition, A ratio T rich or G ratio C rich, the length of segment and number of repetitive units, the satellite DNA is classified into many categories such as microsatellites and metasatellites, etc. These sequences normally do not code for any proteins, but they form a large portion of human genome. These sequences show high degree of polymerism and form the basis of DNA fingerprinting. Since DNA form every tissue such as blood, hair, follicle, skin, bone, saliva, sperm, etc. from an individual show the same degree of polymorphism, they become very useful identification tool in forensic applications. Further, as the polymorphism are inheritable from parents to children, DNA fingerprinting is the basis of paternity testing in case of disputes. As polymorphism in DNA sequence is the basis of genetic mapping of human genome as well as DNA fingerprinting, it is essential that we understand what DNA polymorphism mean in simple terms. Polymorphism variation as genetic material arises due to mutations. Recall different kind of mutations and their effects that you have already studied in chapter 4 and in the preceding sectors in this chapter. New mutations may arise in an individual either in somatic cells or in the germ cell cells that generate gametes in sexually reproducing organisms. If a germ cell's mutation does not seriously impair individual's ability to have offsprings, how can transmit a mutation, we can spread to the other members of population through sexual reproduction. Allelic. Again, recall the definition of alleles from chapter 4. Sequence variations that traditionally been described as a DNA polymorphism if more than one variant allele at a locus occurs in human populations with a frequency greater than 0.01. In simple terms, of an inheritable mutation is observed in a population at a high frequency. It is referred to as DNA polymorphism. The probability of such variation to be observed in a non-coding DNA sequence would be higher, as mutation in these sequences may not have any immediate impact in an individual's reproductive ability. These mutations keep on accumulating generations after generation and from one of the bases of validity and polymorphism. There is a validity of different type of polymorphism ranging from single nucleotide change to very large scale changes. For evolutions and speciations, such polymorphism play very important role, and you will study these in detail in higher classes. The technique of DNA fingerprinting was initially developed by Alice Will Jeffrey. He used a satellite DNA as probe that shows very high degree of polymorphism. It was called a variable number of tandem repeats, VNTR. The technique, as used earlier, involved southern blot hybridization using radioable VNTR as probe. It included first, isolation of DNA. Second, digestion of DNA by restriction endonucleases. Third, separation of DNA fragments by electrophoresis. Four, transferring blotting of separate DNA fragments to synthetic membranes such as nitrocellulose or nylon. Fifth, hybridization using labeled VNTR probes. Sixth, detection of hybridized DNA fragments by autoradiography. A schematic representation of DNA fingerprinting is shown in figure 5.16. The VNTR belongs to a class of satellite DNA referred to as mini satellite. A small DNA sequence is arranged randomly in many copy numbers. The copy number varies from chromosome to chromosome in an individual. The numbers of repeat show very high degree of polymorphism. As a result, the size of VNTR varies in size from 0.1 to 20 kilobase pair. Consequently, after hybridization with VNTR probe, the autoradiogram gives many bands of differing sizes. These bands give a characteristics pattern for an individual DNA, figure 5.16. It differs from individual to individual in a population expect. In the case of monozygotic identical twins, the sensitivity of the technique has been increased by use of polymerase chain reaction PCR. You will study about it in chapter 9. Consequently, the DNA from a single cell is enough to perform DNA fingerprinting analysis. In addition to application in forensic science, it has much wider applications such as in different population in different population and genetic diversities. Currently, many different probes are used to generate DNA fragments. Summary: nucleic acids are long chain polymers of nucleotide. While DNA stores genetic information, RNA mostly helps in transfer and expression of information. Though DNA and RNA both function as genetic material, but DNA being chemically and structurally more stable, it is better genetic material. However, RNA is the first to evolve and DNA was described from RNA. The hallmark of the double-stranded helical structure of DNA is the hydrogen bonding between the bases from opposite strand. The rule is that adenine pairs with thymine through 2 hydrogen bond and guanine with cytosine through 3 hydrogen bond. This makes one strand complementary to the other. The DNA replicates semi-conservatively. The process is guided by the complementarity hydrogen bonding. The segment of DNA that codes for RNA may in a symplectic term can be referred as gene. During transcription also, one of these strands of DNA act as template to direct the synthesis of complementary RNA. In bacteria, the transcribed mRNA is functional, hence can directly be translated. In eukaryotes, the gene is split. The coding sequence exons are interrupted by non-coding sequences introns. Introns are removed and exons are joined to produce functional RNA by splicing. The messenger RNA contains the base sequences that are read in a combination of three to make triplet genetic code to code for an amino acid. The genetic code is read again in the principle of complementarity by tRNA that act as an outer molecule. There are specific tRNA for every amino acids. The tRNA bind to specific amino acid at one end, pairs through hydrogen bonding with codes on mRNA through its anticodon, the site of translation protein synthesis on ribosomes, which bind to mRNA and provide platform for joining of amino acids. One of the rRNA serve as a catalyst for peptide bond formation, which is an example of RNA enzyme ribozyme. Translation is a process that has evolved around RNA, indicating that life began around RNA. Since transcription and translation are energetically expensive processes that have to be tightly regulated, regulation of transcription is the primary step for regulation of gene expression. In bacteria, more than one gene is arranged together and regulated in units called as operons. Lac operon is a prototype operon in bacteria which codes for gene responsible for metabolism of lactose. The operon is regulated by the amount of lactose in the medium where the bacteria are grown. Therefore, the regulation can also be viewed as regulation of enzyme synthesis by its substrate. Human Genome Project was a mega project that aimed to sequence every base in human genome. The project has yielded much new information, many new areas avenues have opened up as a consequence of the project. DNA fingerprinting is a technique to find out variations in individuals of a population at DNA level. It works on the principle of polymerism in DNA sequences. It has immense application in the field of forensic science, genetic biodiversity, and evolutionary biology.